let us see about the Operon model for transcriptional regulation. The basic concept for the way transcription is controlled in bacteria is called the Operon model. The Operon model was proposed by Francois Jacob and Jacques Monod in 1961 on the basis of their study on the inducible system for the synthesis of beta galactosidase enzyme in E. coli. The Operon model has been variously modified ever since it was originally proposed by Jacob and Monod. In its original form, Operon model assumed the presence of a unit of coordinated control of protein synthesis, and this unit was called Operon as seen in this figure. An Operon consists of 1. An operator gene which control the activity of genes, 2. a number of structural genes which take part in the synthesis of proteins. This means that the structural genes will synthesize mRNA under the operational control of an operator gene. The operator in its turn is under the control of a repressor molecule synthesized by a regulator gene which is not a part of the operon. The mechanism of regulation of protein synthesis utilizing Operon model can be illustrated using the example of two different operons, LAC as well as tryptone operons in bacteria. The LAC operon in E. coli. The LAC operons as seen in this figure consist of 26 base pair. There are 3 structural genes in LAC operon, which includes 1 LAC-Z consisting of 3063 base pair, which codes for the enzyme beta galactosidase, which is active as a tetramer and breaks lactose into glucose and galactose to be utilized in the cell. 2 LAC-Y consisting of 800 base pairs, codes for beta galactose permease, which is a membrane bound protein and helps in transport of metabolites, and 3 LAC-A consisting of 800 base pairs, which codes for beta galactose transacetylase, an enzyme that transfers an acetyl group from acetyl coenzyme A to beta galactosides. Any mutations in LAC-Z and LAC-Y genes give LAC- genotypes which cannot utilize lactose, while LAC-Z- mutants abolish enzyme activity. LAC-Y- mutants cannot take up lactose from the medium. No defect is observed in LAC-A- mutants. It is possible that acetylation gives an advantage for utilizing certain non-metabolizable analogues for beta galactoside because it results in detoxification and excretion. In LAC operon of E. coli, repressor is synthesized through activity of a gene LAC-I known as regulator gene. The repressor then attaches on operator gene and checks synthesis of mRNA. This is clearly depicted in this figure. If lactose is added, the repressor is rendered inactive, so that it cannot attach on operator gene and the synthesis of mRNA takes place. This is illustrated in the figure. The activity of the regulator gene is controlled by a promoter gene on its left side. It has been shown that mutations in this promoter may increase the quantity of mRNA for repressor. Lactose is not the real inducer of LAC operon. Since in the repressed state of LAC operon, E. coli cells will not synthesize beta galactoside permease, one can question how lactose enters the cell. It is believed that a minimal amount of enzyme is always present in the cell to start the metabolic pathway. It has been shown that lactose is not the real inducer of LAC operon, but rather it is an anti-inducer, since it binds to repressor to increase its affinity for operator. On the other hand, the bound protein in inactive repressor was allolactose. It has also been shown that while beta galactosidase breaks lactose into glucose and galactose, a side reaction changes galactose into allolactose and galactobiose. Therefore, in the regulation of lac operon, lactose is taken up and a part of it through galactose is converted into allolactose, which then induces the operon and outweighs the anti-inducing effect of lactose. Let us see about constitutive mutants. It has been found that a mutation in regulator gene LAC-I results in the absence of active repressor and consequently results in uncontrolled synthesis of enzyme. Such strains which continuously synthesize enzyme independent of need are known as constitutive strains or constitutive mutants. These constitutive strains can also be obtained by mutations in the operator gene. A mutation in operator gene does not allow repressor to act because the repressor functions by acting on operator site. Therefore, there are two types of constitutive strains, one regulator constitutive and two operator constitutive. The above two types of mutations, though have the same net result, but differ in their location as well as in their dominant recessive relationship with respect to the wild type. This relationship has been worked out by using the partial diploid cells, merozygotes. Merozygotes are those which have one complete chromosome and a part of another chromosome covering the desired region. When heterozygotes for regulator constitutive were derived, no enzyme synthesis was observed, suggesting that wild LAC-I is dominant over mutant LAC-IC. This is illustrated in this figure. On the other hand, partial diploid cells heterozygous for mutation at operator site exhibited constitutive synthesis of enzyme, indicating that normal operator gene O is recessive to mutant gene OC. This is clearly depicted in this figure. There is yet another class of regulator constitutive mutants which are dominant LAC-ID, so that in merozygotes transcription continues. This LAC-ID mutant gives repressor subunit defective at N-terminal region. This defective subunit takes part in formation of a tetramer, which is partly derived from LAC-I and partly from LAC-ID, so that the tetramer is unable to bind on the operator.